Yes, I'm here. Thank you, Salman. So good evening or good afternoon or even good morning, wherever you're based. Train is interested in the program. It's cool for me to speak last because I'm going to have a very short introduction on our research interest. You've now heard that many faculty affiliated with the BME department at UIC are interested in the matrix, in the extracellular matrix, and that's also the focus of my lab. But we do things slightly differently because I didn't train as an engineer. I'm a cell biologist by training, and so I come from that perspective talking to you today. As you probably all know, the extracellular matrix is a fundamental component of multicellular organisms. It organizes tissues, it organizes organs, and you're, I'm sure, all familiar with its architectural and physical roles. But the extracellular matrix is much more complex than that, and it actually sends signals to the cells and govern all cellular processes. So the matrix can tell whether a cell should divide or not, should differentiate or not, should remain a stem cell or not, should survive or not, or in the context of disease such as the one we study, which is cancer, whether a tumor cells can become migratory, invasive, aggressive, and form metastasis. So the matrix plays key roles in health, in development and health, but also in diseases. Yet, there is very little known about which proteins compose the extracellular matrix. And in fact, even if you're not an ECM specialist, you can appreciate that the matrix of a bone is going to be different than the matrix of the brain or different of the matrix of the liver, right? These tissues have different mechanical properties and function, and so their matrix is going to be different. And yet, we don't know how different they are. Next slide. So what my lab does is develop proteomics approaches and computational approaches to profile the protein composition of the extracellular matrix. And I'm not going to enter too much into the details, but over the years, we've built several pipelines, allowing us now with high throughput to profile in a very unbiased manner, right? We're not making any assumption which proteins are present. And usually what we do is compare two different conditions. So again, my lab studies extensively cancer, but also fibrosis, like Jaewon Shin's lab. And one of the questions we ask is, well, how does the matrix of a normal lung differ from the matrix of a lung fibrotic tissue? How does the matrix change between a poorly aggressive tumor versus a highly aggressive tumor? And when we do that using proteomics and bioinformatics, we can then identify ECM signatures characteristic of a disease state. Now, because we're biologists and in fact, we're based in the College of Medicine, we don't stop there. We follow these screens by what we call mechanistic studies or interventional studies when we're going to try to dissect the role of the proteins we've identified as differentially expressed on the process we study, for example, cancer. And so we're going to either do gene editing, pharmacological inhibition, and then use both cell culture system, but also animal models in the context of preclinical studies, again, to dissect the role of the proteins we will have identified by proteomics. And then if we're successful, our hope is to contribute to identify two things: novel diagnostic and prognostic markers of certain disease states. And we've shown that we can do that in the context of breast cancer, colorectal cancer, and skin fibrosis. But we're also hoping to be able to identify novel therapeutic targets in the extracellular matrix environment that then we can act on to prevent the progression or even development of certain diseases. Next slide. So I'm an assistant professor. I've had my lab for almost 6 years now at UIC. So my lab is fairly young, but I've already graduated one student in 4 years, and the second one is about to graduate. And I'm also placing undergrads and grad students, both in the academic sector, but also in the private sector. And in fact, we have quite a few collaborations with big pharmaceutical companies listed on this slide. My lab is really the lab pioneering extracellular matrix proteomics that puts us in a cool position to really develop breakthrough technologies and allowing to break new grounds in the field of ECM research, and that's pretty cool. But we're also at the intersection of many different disciplines. I've talked about big data and proteomics and computational biology, but I'm, again, trained as a basic cell biologist. And so we bring things together. And what I'd love to do in the near future is expand further my lab to bring in people who have a more engineering perspective on the study of the extracellular matrix to keep on advancing our research program. I'm very big on mentoring and career development, and I'd be more happy to tell you all about that if you're interested in contemplating maybe joining my lab, either for a master's program or a PhD program. And I know Salman emphasized the importance of being funded. It's true, if you're going to join the lab, you want to make sure you can be paid for the work you do, but also have enough money to do all the crazy experiments you're going to be thinking about. And we're in a fortunate position to be well funded. We certainly work hard, right? And we do really cool things in the lab, but we like to have fun outside the lab. And so I thought I'd put here a picture of our latest summer lab outing, where, we took the lab and went to the Ice Cream Museum of Chicago. For those of you not based in Chicago, Salman was saying that Chicago is a really cool city. Well, we now have an ice cream museum, which was a perfect place to relax over the summer and celebrate graduation and publications and successful grants ahead of the start of the new academic year. Next slide. These are my contact infos. We have a website, the lab's on Twitter if you want to follow the daily activities and exciting news that we're sharing. As Salman explained, we're going to follow the process of contacting me with certain research interests and specifics, contacting the program directly. If some of you are interested in exploring the extracellular matrix, you don't need to have studied it before. That's something I can teach you, but think about what you could also bring to my lab with this engineering experience. Maybe some of you are into computational biology or bioinformatics, maybe math or statistics. I'm looking to diversify the expertise of my lab, and so I'd be very excited to bring on board engineers to help us advance our research.